He's not coming back.
Yeah, I think we are all here. Good morning, everyone, and welcome to this session that is focusing on the latest advancements in fast virology. I'm Kirill Krstevsky, and I'm very pleased to be your moderator during these 2 hours. Today, we will begin by exploring the complexities of foot and mouth disease viral infection in different susceptible species, and we will be challenging some long-standing assumptions in the FMD community. These insights remind us how critical it is to adapt our approaches based on species-specific responses. From there, we will look at the mechanisms that allow FMD virus to persist and innovative models that provide new ways to study the convalescent and persistent phases of infection. We will then pause for 15 minutes and we'll welcome questions from the audience that the first 4 speakers will respond. Next, in our second part, we will hear about low-cost methods to inactivate FMD virus in field conditions before shifting our focus to lumpy skin disease and get new insights on transmission pathways of lumpy skin disease virus that are crucial for refining our prevention and control strategies. Together, these talks offer a broad view of the dynamic nature of foot and mouth disease and lumpy skin disease virus research. Thank you for joining us, and let's start with our first presentation. Speakers, please be informed that you have 8 minutes for your presentations. Our first speaker in this session is Michael Eschbaumer from the Friedrich Loeffler Institute. Michael has been head of the German National Reference Lab for FMD since 2016, and he is a member of the EU FMD Special Committee on Biorisk Management. His research interests are safer and more effective FMD vaccines, persistent FMD virus infection of ruminants, intra- and interspecies recombination of aftoviruses, and ways to improve biosafety of FMD sample collection, transport, and diagnostic testing. Today, Michael will be presenting a presentation called Cautionary Tales of Intra-oropharyngeal Inoculation of Pigs with FMDV. Michael, please.
Yes, thank you, Carol. Usually when you see the logos of different institutions on the same slide, it's because it's a collaborative study of some sort. Sadly, this one was not. It's not that we came together to do this thing, it's just that after the fact we found out that we were all trying to do the same thing and kind of had a similar experience with it. That's why we're now all mentioned here. The whole story of experimental FMDV infection of pigs, it's an old one. I mean, there's a good old gold standard method that has been used for a long time. It's still commonly used, for example, for vaccine trials, where you just take the virus and you inject it into the heel bulb of the pig. Works very well, works reproducibly, but for some applications, it's considered a bit too artificial, because obviously by taking the virus with a needle, sticking it in the pig, you're bypassing a lot of the natural physical barriers and the natural immunity that is involved in an actual FMDV infection out in the wild. So to kind of go to the other end of the spectrum of how close you are to what is actually happening in nature would be contact exposure. So you have pigs that have been infected with FMDV, for example, by needle, and then at some time after their inoculation, when they start developing FMD, when they start shedding the virus, you bring them in contact with the other pigs that you want to be infected as part of your study. It's close to what's happening out in the wild, but at the same time, you kind of lose control over how much virus is actually being passed on to these in-contact pigs. And if you really want to have like a defined dose, there are concerns that what you're giving the pigs here is a lot more than what you would usually apply, for example, by injection or other means. So kind of in between of these two things, something that is artificial, but maybe not too artificial, is this whole idea of the intra-oropharyngeal inoculation, where you combine the near-natural exposure on a mucosal surface with full control over how much of the virus you give, when you give it, etc., which makes it a good choice for something that tries to look at more the natural processes of FMDV. How does that work? This is a technique that's been pioneered by a group from Plum Island. Carolina Stenfeldt, the speaker after me, was spearheading this effort. You just see some pictures on the left here. You lay the pigs on their back. You put the virus on the tonsil of the soft palate, right there in the back, and then you leave it there for a while. Different people do it differently. Either you then afterwards remove the inoculum, or you leave it in place as in the original method, and you really try to keep it in contact with the tonsil itself. Before that, keep in mind that you're not scratching the epithelium or anything. You're really just putting it in there without any intentional epithelial injury. We tried to do this at FLI. People did it at Purbright. People did it in Wageningen. With different viruses, different doses of different doses of virus, from the kind of small to what I at least thought was high to excessive. And yeah, you're going to see that our success rate with this was at this point not very high. We also then, and this is why it's highlighted here, we went to the O-Taiwan 97 out of concerns. Oh, maybe we need a really pig-adapted virus. Maybe this is kind of like an interspecies different thing, as has been mentioned. So we went for what is probably the most pig-adapted virus out there to include it in this as well. And for comparison to the intra-oropharyngeal inoculation, we also, in some of these experiments, gave the same amount of virus by injection into the heel bulb with generally lower doses. And then we looked at the pigs and see what happened. But as I said, in about half of the experiments that we did, even when you put in very high doses, in excess of 10 to the 6 or 10 to the 7, in a group of 5 to 6 pigs, you would end up with maybe one pig actually being infected by the inoculation. As you can see here, number 15, where you get onset of disease fairly early on after inoculation, and then thereafter you see the virus spreading within the pigs that are kept in the same room, but there's always this delay indicating that they didn't get it from us, but instead they got it from their buddy that had gotten it from us. And then there were other experiments where a similar dose of virus or even higher doses of virus were used, and we didn't even get this one initial infection. So there was this clear problem with getting the initial animal infected, which was not there at all when we did the needle inoculation. There, even at lower doses, you consistently got disease and got it very quickly. So I'm not up here to really trash talk work that has been done by other people. There's a lot of good papers out there where IOP works really well. It works really well in Plum Island. There's a group in Japan. I think some members of this group are also in attendance. There, the IOP method also worked really well. So for me, the question really is, what are we doing wrong here? Because it shouldn't be so hard to infect pigs with FMD. But it may, and I'm really hoping for input from you more than anything else. We did see that in the few cases where we manage to successfully infect a pig in the room, the virus then spread quickly. So it's easy for a pig to infect another pig. It may just be a little bit harder for a human to infect a pig in the first place. And we have to look at things like, are there virus-specific differences? Are there like tiny differences in how the method is being applied that then kind of push it over the brink where you do not get a good initial efficacy? And I also have to say that Carolina and her group have been very helpful in they've come to our place now and we've done it together, and we're gonna look at really try and get to the bottom of how this works. But at the moment, really, as I put in the title, this is more of a cautionary tale. If you want to do intra-oropharyngeal infection of pigs, basically anticipate that it may not be as quickly and as easily as you think it would be, and try to really get to the bottom of how pigs get infected and what we need to do to make sure that we can reliably infect them in a laboratory setting. And so in the panel discussion, I was hoping to really hear from you, from your experience. Have you done experimental FMDV infections? What routes have you used? What outcomes have you gotten? What viruses have you used, etc.? Thank you.
Thank you, Michael. Thank you very much, Michael. And as I said, please make notes of your questions, and Michael will be, and all the other speakers will be able to answer them in the questions and answer sessions. Our next speaker is Carolina Stenfeldt. Carolina Stenfeldt is a scientist working at the Foreign Animal Disease Research Unit of USDA, ARS at the Plum Island Animal Disease Center through a collaboration with Kansas State University. Her research is focused on investigating FMDV pathogenesis and transmission in different livestock species with a specific interest in the FMDV carrier state. The title of Carolina's presentation for today is "Sheep are not little cows when it comes to FMD." Very interesting. Carolina, the floor is yours.
It should be obvious, shouldn't it? Thank you for that. So I work for both USDA and Kansas State University in the lab that is run by Dr. Jonathan Arzt, who unfortunately couldn't be here today. But we specialize in studying FMDV pathogenesis and transmission in different hosts. And through the past year and a half or so, we've been focusing mainly on sheep. So today, I just want to give kind of a brief overview of some key concepts that we've learned. I only have 8 short minutes for this. So there won't be that much detail, but I want to provide a summary of what I would consider like the bare essentials that you need to know if you want to work with foot and mouth disease in sheep, whether it be in the lab or in the field. And so the 3 main thematic points I want to touch upon is on basic anatomy, clinical presentation of FMD in sheep, and optimal sampling for detection of FMD in sheep. And so the main emphasis to this will be to focus on what's different in sheep compared to primarily cattle. So this might not be like groundbreaking science, but I think there are some important aspects here that are often overlooked when people go from one species to the next. So starting with the basic anatomy, the upper respiratory and gastrointestinal tracts of the sheep are very similar to cattle. So we have the nasopharynx, that is part of the respiratory tract that connects the nasal cavity to the trachea. We also have the oropharynx, which is a distinct compartment. It's basically an extension of the oral cavity that when the animal swallows and the soft palate gets lifted out of its resting position, provides passage to the esophagus. When it comes to FMDV in sheep, the upper respiratory tract, so the nasopharynx, is extremely sensitive to virus exposure. That's very similar to cattle. And that represents the initial site of infection. But quite early in the infection, there's also dissemination of virus to the epithelial components of the local tonsils in the area. An absolutely critical difference from cattle is that in sheep, FMDV primarily persists in the epithelial crypts of the oropharyngeal tonsils. So now we're back in the upper gastrointestinal tract. And that involves both the palatine tonsils and the paraepiglottic tonsils, which are tiny little tonsils of the side of the epiglottis that people don't really notice. But they're important for FMDV infection, and we find a lot of persistent FMDV there. So, and I'm going to get back to why that is relevant in a few slides, but I want to first move on to the slightly contentious topic of clinical FMD in sheep. Now, sheep actually do get clinical FMD, but it often looks a little bit different compared to what we are used to seeing in the other hosts. The most common signs we find are high fever and lameness on one or more feet, but that lameness is not necessarily associated with easily identifiable vesicles. What you will often find instead is increased temperature in one or more hoofs, and also some deep edema around the coronary bands. This colloquial term, puffy coronary bands, was coined by one of our postdocs, and I actually find that quite descriptive, as you can usually feel this with your hands easier than you can see it with your eyes. What is very important and absolutely different from cattle and pigs is that there's typically quite a lot of within-group variation. So you can have a group of similar sheep that are all infected with the same virus. Some of them will be very severely affected. Others will walk around, look pretty much unaffected. But those animals are typically also infected, and they are shedding about equal amounts of virus in secretions as the animals that are clinically sick. I want to show just a few examples of lesions in sheep. These are coronary band vesicles. These are actual vesicles. These are not the edematous coronary bands I mentioned before. But what's specific here, and that I find is quite common, is that these coronary band vesicles are located beneath the upper portion of the hoof wall. These are very painful. The animals are very lame. They do have lesions, but these don't really look like the way we're used to seeing common FMD vesicles. And it's essentially impossible to get an epithelial sample from these vesicles because they're underneath the hoof wall. Some sheep are a little bit more conventional and get more conventional looking FMD lesions, typically in the interdigital cleft. My experience is these are often a little bit later in the clinical phase of disease compared to the other lesions, but they're definitely more easily identifiable and clearly also easier to get a sample from. I want to touch just briefly upon different sample types that we use to detect FMDV in sheep. Our favorite sample type for detecting infection during the early phase of infection are these oropharyngeal swabs. So they're these large cotton swabs. They're actually gynecological swabs, so they should hopefully be available in many places. We target the oropharynx, so basically as far back as you can get through the oral cavity with the dry swab. We then insert the swab in media, and when we get back to the lab, we centrifuge it to get the liquid back out of the swab. These are very good for both PCR and virus isolation for approximately 2 weeks after infection. They're not good for consistent detection of persistent FMDV. For that, you need to reach for your probank sample, but note that the probank that we use for sheep is not the same as for cattle. This is actually not a cup at all, and you can cause quite a lot of injury to sheep if you try to use a cattle probank. If you remember what I said in the beginning, FMDV actually persists in the oropharynx of the sheep, which is different from cows. If you want to collect a probank from a cow, you would insert the probank up through the mouth and wait for the cow to swallow, because that lifts up the soft palate and provides access to the dorsal portion of the soft palate and parts of the nasopharyngeal mucosa, which in a cow is what you want to sample. But in the sheep, I find we get better consistent detection if we avoid going too far in and instead focus on scraping the sort of dorsolateral aspects of the oropharynx, and then clearly rinse your tool in probank. In reality, it looks something like this. It can be somewhat useful to estimate the distance to the eye on the outside of the animal before you sample, and then try to knock it further back in that. Now, sheep are not little cows, and they're quite different here in that they absolutely hate probank sampling, and they'll put up quite a bit of resistance to it. Partially based on that, we wanted to try an alternative approach for detection of persistent FMDV. So this method is based entirely on what was published for African buffalo by Brian Charleston's and Francois Murray's group. So we used this human cytology swab and a laryngoscope to actually access these palatine tonsil crypts that are in the very back of the oropharynx. You need to actually get into the crypts. It's not enough to just swab the oropharynx. And this is very easy to do if your animal is sedated. It's quite difficult to get your swab into those crypts if the animal is not still. But it was an interesting concept, and it provided some pretty graphs, which I want to show some examples of here. This is FMDV detection in an experimentally infected sheep for 40 days. We have detection in serum and these oropharyngeal swabs in red and green on the left-hand side, and on the right-hand graph side of the graph is the persistent infection, when we only have detection in the probank samples and these tonsil crypt swabs. They're approximately equal. So, the main conclusion, sheep do get foot and mouth disease. There's definitely greater within cohort variation in clinical signs, and the clinical signs might look a little bit different compared to what you're used to seeing, but they are often present. Very relevant is that the anatomic site of FMDV persistence is different from cattle, and you want to be aware of this and adapt your sampling technique for optimal detection of persistent infection. So, as I mentioned, this work was done in the lab that's run by Jonathan Arzt, and clearly several other people involved, so thank you for everybody who supported this work, and thank you all for listening.
Thank you very much, Carolina. Our next speaker is Sandra Blaise-Boisseau. Sandra is Deputy Head of the Biopic team of the ANSES Virology Unit, which is in charge of the mandates of the National Reference Laboratory for FMD, SVD, VSD, VOA Reference Laboratory, FAO Collaborating Center, and EU Reference Laboratory for FMD. She is leading research projects focused on host FMDV interactions, aiming to elucidate mechanisms of FMDV persistence in ruminants and contribute to better understanding and control of FMD in livestock. The title of Sandra's presentation is "The Leader Protein is Necessary for the Establishment Persisting Infection by FMDV Type O." Sandra, the floor is yours.
Thank you. Good morning, everyone. And thank you to the scientific committee for giving me the opportunity to present this collaborative work. And this collaborative work is take part of better understanding of FMDV persistence mechanisms that remains an obstacle to FMD control. So, more precisely, the context of the study, one of the aims was to uncover alterations of host response during persistent FMDV infection in cattle. And to this aim, we focused on the leader protease, and this is the major viral protein known to be involved in host innate response evasion during acute infection. It inhibits host cell mRNA translations and blocks interferon response in vitro. And previous work have shown that leaderless FMDV variants of serotype A were strongly attenuated in vivo in swine and cattle. And the question was, could leaderless FMDV establish a persistent infection? So the first work was to produce leaderless virus derivative of the O France 1 2001 virus. And it was done by deletion of the LB sequence. And first work was the study of growth kinetic. It was done in interferon incompetent cells like BHK21, and we observed similar growth of both viruses, mutant and wild type. But in the interferon competent cells, we observe a clear disadvantage of the leaderless mutants in the fitness. So to further study, we use this model of primary bovine cells derived from the nasopharynx, so dorsal soft palate, so DSP, and grown at the air liquid interface. And we perform this assay until 35 days post-inoculation with MY 0.01, inoculating wild-type virus or the leaderless one. And we sample every 2 or 3 days the cultures. So, what about the results? No persistent infectious leaderless could isolate it, even on sensitive BHK interferon incompetent cells. We only detected residual leaderless viral RNA, and only until 28 days post-inoculation. And concerning the viral loads, estimated with titer TCID 50 titrations in BHK cells, we observed that the leaderless virus was undetectable after the early stages of infection. We also started to conduct transcriptomic analysis on the DSP cells, and the first results indicate that many differential host genes are differentially expressed between leaderless of wild-type infections at 12 hours post-inoculation. So this work was completed with an in vivo study conducted in the Michael labs at FLI, and a simulated infection route was done in two groups of cattle using the intra-nasopharyngeal route. And the assay was conducted until 35 days post-inoculation, and the animals were sampled until 35 days, then euthanized. But 2 animals per group were euthanized at 24 hours post-inoculation. So as you can see, concerning the remaining wild-type group, all animals remaining developed vesicular lesions beginning on 4 days post-inoculation, but in the leaderless infected cattle did not develop any vesicular lesions of clinical sign of FMD. About the virus shedding, so virus shedding in nasal samples was confirmed just in samples taken just after the inoculation, but then we observe only a peak of shedding between 6 to 10 days for the wild-type group, and 2 persistently infected animals were detected, so incidence of around 33%. And while for the leaderless group, no viral RNA was detected in any samples in the course of experiments. So, in conclusion, we observe that the results obtained in vitro and in vivo are in line. So in the DSP-Ali model, we observe that the leaderless virus O was strongly attenuated, and this virus cannot establish in our model, cannot establish a prolonged nor persistent infection. And we are in progress to understand better the role of L-pro-mediated immune evasion in persistent FMDV infection in this model through IPR results pending on early time points samples, so 6 and 12 hours post-inoculation. And for the in vivo experiment in cattle, as shown, the leaderless virus doesn't trigger any clinical signs and any detectable virus in serum, secretions, or tissues in this group. Incidence of persistently infected animals in the wild-type group was around 33%, so it is in the mean range, but it can be modulated because of the small effective of the group. And overall, we can say that this confirms the strong attenuation of leaderless FMDV for a new serotype and a virulent panzootic parental lineage. And these leaderless viruses could be used for safer production of inactivated vaccines or for virus neutralization tests with reduced biocontainment requirements. So, thank you for your attention, and thank you for all contributors, partners, and beyond in this project.
Thank you, Sandra. Thank you for sharing your results with us. Our next speaker is Anne-Sophie Bultinck. She will present on exploring the guinea pig model in the convalescent and putative persistent phase of infection with FMD virus. Anne-Sophie Bultinck is junior scientist at the Laboratory of Exotic and Vector-Borne Diseases at Cienzano. In 2023, she obtained a master's degree in biomedical sciences from Ghent University, where she gained experience in conducting experiments with laboratory animals. At Cienzano, she investigates the potential of guinea pigs to study the convalescent and persistent phase of infection with foot and mouth disease virus. And Sophie, the floor is yours.
Thank you. Hello everyone, and thank you for giving me the opportunity to present this work. Together with David Lefebvre, my colleague, I've been researching the exploration of the guinea pig model in the convalescent and putative persistent phase of infection with foot and mouth disease virus. First of all, some background on persistence of FMDV in natural hosts. Persistence refers to the virus's ability to remain within the host for extended periods post-infection without causing any overt clinical signs. So it is defined as animals being virus positive for at least 28 days post-infection. The persistent FMDV virus is mostly located in pharyngeal tissues, and animals in which persistence has already been observed include cattle, African buffalo, sheep, goats, and wild ungulates. The persistent phase can last for months to even up to years, depending on the species. And the importance of persistence is that persistently infected animals play a role in maintaining the virus within a population. However, this does not mean that there is always active viral shedding or transmission. And there is limited evidence of persistence of FMDV in guinea pigs. Now, why do we use guinea pigs? Previous research has already shown that guinea pigs are a suitable animal model to study the acute phase of FMDV infection. Why is that? Because when inoculated with FMDV, they show similar vesicular disease as observed in natural hosts, which means vesicular lesions in the foot pads and in the mouth and tongue. This acute phase FMDV guinea pig model is already used to study antivirals and vaccines. And guinea pigs offer several advantages, for example, financially, but also logistically, it is easier to house guinea pigs than natural hosts, and the risk biosafety is also less. Previous research at our lab has already established an acute guinea pig model for FMDV. In this study, guinea pigs were inoculated with a guinea pig adapted strain of FMDV, namely the O1 Manissa Turkey strain. And here they saw that guinea pigs developed vesicular lesions in both the hind foot pads, which is the inoculation site, but also the front foot pads and the mouth and tongue, already starting at 2 days post-inoculation. And these vesicular lesions remained visible until day 8. However, by day 8, they started to heal. Then several samples were also collected from these guinea pigs, and by 3 days post-inoculation, these were positive for FMDV virus. These samples include serum, oral swabs, foot pads, tongue, but also internal organs. But as you can see, this viral load also diminished quickly because by day 8, only 1 guinea pig remained positive in serum and oral swab. Now, what is our aim of our study? We want to enhance the knowledge on the pathogenesis of FMDV in guinea pigs. And what we are doing differently is that we also look at later phases of infection. So we look at 14 days post-inoculation, which is the convalescent phase, and 28 days, which is the putative persistent phase. And the ultimate goal is to then develop a guinea pig model for persistence FMDV, on which we can test antivirals and vaccines, and thereby hope to find a way to prevent or overcome this persistence. How did we do this? We inoculated guinea pigs intradermally into both hind foot pads with the FMDV O France 1 2001 strain. We specifically chose this strain because experimental infections have already shown that this strain causes a high percentage of persistence in natural hosts and can also persistently infect cell models. The guinea pigs were then examined daily for clinical symptoms and body weight, and were then euthanized at the different time points. And I see that the names of the phases have switched. So we euthanized the guinea pigs on 3 days post-inoculation, the acute phase, 8 days, the subacute phase, and then also 14 and 28 days, the convalescent and putative persistent phases. Then, from these guinea pigs, samples were collected and tested with RT-qPCR for viral load. The results of the acute phase, then, I highlighted 3 different samples, namely the serum, spleen, and liver, because these showed the most interesting results. What did we see is that for half of the guinea pigs, in these 3 samples, a high viral load was present, while for the other half of the guinea pigs, a lower viral load was seen, or even an absent viral load. You can also see this in the figure that each dot is a guinea pig and it's divided into 2 groups. For the guinea pigs that had a higher viral load in serum, spleen, and liver, these also had detectable viral RNA in the heart, the lungs, the front foot pods, the bronchial and abdominal lymph nodes. However, important to mention is that in both groups, actually, no vesicular lesions were observed in the hind foot pods or the front foot pods or the tongue. And also between both groups, there was no apparent difference in viral load in the hind foot pods. And both groups did not show any clinical signs or did not differ in body weight. Then the results at later phases of the infection, I highlighted the popliteal lymph nodes here because these showed the most interesting results. The popliteal lymph nodes are the lymph nodes draining the inoculation site, which are the hind foot pods. And here, viral RNA was consistently detected up until 28 days post-inoculation in all guinea pigs. And interestingly here is that on day 14, there appears to be an increase in viral load in these popliteal lymph nodes. And this was only in the popliteal lymph nodes and not in the hind foot pods. What can we conclude from this? First of all, FMDV O France 2001 appears to have a tropism for blood, spleen, and to a lesser extent, the liver. And previous researchers have also seen this in laboratory mice, where FMDV was trapped and retained in the spleen for up to 28 days post-infection. Secondly, there appears to be a role for the popliteal lymph nodes, and this was also seen by other researchers in pigs, where viral RNA of FMDV persisted in lymphoid organs, draining previous lesion sites even beyond clearance of infectious virus. I hope everything was clear, and thank you for listening.
Thank you, Anne-Sophie. And thanks to all our speakers for respecting the time allocated for their presentations. Now we can open the question and answer session, and I invite the audience to ask their questions. Please.
Thank you. I have a question regarding the inoculation route on pigs. Because it's a little surprising to me, the level of, I don't know if it's myocarditis, what you identified in the pigs that were dying by fulminant death of the animals. You mentioned that was a finding on your experimental design or the result based on the route that was utilized.
That's interesting that you asked that, because actually in that presentation I didn't mention myocarditis at all, well, at least not intentionally. The pigs that we had here that were infected by like horizontal transfer from the inoculated pigs in the room, they did get severe FMD, but that was mostly lesions on the feet. Like they were extremely lame. They couldn't walk anymore, and we euthanized a lot of these pigs just on that account. So at least in our hands, in these experiments that I presented here, we didn't have any like sudden death from myocarditis. But that being said, we have other more recent data that I have not talked about here yet, where we did see some acute deaths also after IOP.
So do you think it's the route? Because we have done contact and also the direct inoculation in the heel bulb, and we don't see evidence of that fulminant death.
Carolina is the myocarditis specialist, so I would defer to her on that.
Yeah, before last week, I only had one pig with myocarditis in 12 years, and then last week we had 3. So I don't think it's the inoculation route.
But then what is it?
Okay, thank you. We have another question over there.
Hi, question for Anne-Sophie about is there a requirement for your virus to be guinea pig adapted? And I wondered if you had sequenced your viruses that are in the animals where the virus is replicating to a much higher level. Is there any difference that's happened in those viruses?
So to answer your first question, we now chose a strain in which we saw that it can persistently infect natural hosts and cell models, but over the past month we have also done a new experiment with the guinea pigs in which we used the guinea pig adapted strain. For this I don't have any results yet because it was too last minute, but we did see that this strain caused vesicular lesions, that we did not see with the O France strain. So that might be an advantage of using the guinea pig adapted strain. And then to answer your second question, we did not do any sequencing. However, that might be interesting to see whether this strain changes before versus after inoculation in the guinea pigs. I hope this answers your question.
Thank you. We have another question. Yeah, okay. Okay, first, a colleague in the back, yes, and then we go up front.
Yeah, thank you, presenters. To Anne, I just have a worry. If from your study, the FMD persistence was obvious in lymphoid organs, then what then is the role of humoral immune response in curtailing the pathogenesis of the FMD virus adapted strain you used?
You mean the humoral immune response in the guinea pigs? We have not yet investigated whether these guinea pigs have the humoral immune response. However, previous research has shown that the humoral immune response is increased after inoculation with FMDV. But the role of this humoral immune response in combination with the lymphoid organs is yet to be confirmed.
Okay. So, Michael, yeah, thanks. Your work in terms of the possibility of the route of inoculation, I'm just wondering, did you try to mimic the natural route of infection of the FMD virus in your own model, or there were other reasons why you chose that type or route of inoculation?
Yes. So we
no,
first...
to Michael.
Oh, sorry.
The contact exposure works very well. I mean, it's certain for many applications it does. I think is the best model to use. But in this case, we were looking for something to be a bit more controlled, as I indicated in the introduction, where you really can say, okay, we inoculated this amount of virus at this point in time, so that you can compare between different groups and different viruses. That was really the purpose of using the IOP. In this study we were trying to look at differences in virulence between different genetically modified viruses. That was the original objective, where you would just want to say, okay, we expose these pigs to 10 to the 6 TCID50 of virus A, and these to 10 to the 6 TCID50 of virus B, and we want to see differences in virulence, in viral shedding, what have you. And if you try to design experiments like that using contact exposure, it becomes a lot more difficult or like impossible to really do that. That's our reason to go for IOP here.
Okay, I feel I have a comment here since I kind of invented the method. We know from history that there are this historic events of pigs getting FMD by eating contaminated waste, and it's believed to be a natural route of initial infection in pigs, and that's why we developed the method that way. And we've used it for many years without having these problems. But I want to make a comment of all these, we call them simulated natural methods, which include both this intra-oropharyngeal method in pigs and the intranasopharyngeal that we use very efficiently in cattle and sheep, is that you can't think of dose the same way as you do when you inject the virus. When you inject a virus, it'll replicate at the injection site, and all of your inoculum will enter the animal. What you do in these simulated models is you place your inoculum on a mucosal surface, and even if you put 10 to the 7 logs of virus there, only a very small fraction of that is going to enter any cells, and I would see it more as a probability equation if you have a higher dose, but also a function of the contact duration, and it's a probability of the virus to actually enter the tissue, so you won't see the same dose effects as you would. It's more sort of binary, but more think of it as a probability equation, whether you get infection or not.
We have a question here, then in the back, and we will take the third question.
Thanks, Chair. Nice presentations. One comment and a question. Comment regarding the appearance of myocarditis. We have found that certain strains cause myocarditis, not all strains. We have seen in Myanmar 98 causing myocarditis.
In what species?
In pigs.
In pigs.
In pigs, that too in young pigs, but not serotype A.
Mm-hmm.
That's been our observation. A second question is to you, Michael. What was the type of virus you used? Was it a cell culture supernatant, a cattle-adapted virus, or a pig-adapted virus?
That's a very important point, yes. So these are several rounds of infections that we tried to do. We started out with virus that was created from an infectious clone. So it was tissue culture virus. We saw these isolated positive outcomes with that. And then this was one of our first concerns. Well, maybe it's just tissue culture virus, so it doesn't really work well in a simulated natural approach. So we went in, and from these few animals that did get infected, we collected lesion material, we homogenized it, titrated it, and then went back to do a second round. Same dose as before, but now the pig passage virus, and the outcome was pretty much the same. And then we actually tried it a third time. We then had one pig there that we collected again material from, so now we're looking at 2 pig passages and still similar outcomes.
So did you consider the pH of the oral cavity when you instill the virus there? Because the pH is somewhere around 8.
Thank you, Najenda.
Salivary fluid. So, and again, there has to be a breach in the epithelial surface, especially in that region, to mimic the oral route, because when they eat whatever swill or something, there's going to be some damage that's caused because of the way they eat. So your observations, please.
There's 2 very interesting angles, and you saw from Carolina's reaction, that was really one of the things that we looked at just last week when we were really going over the protocol step by step, looking at what we did and looking what Carolina does. And one difference was that they add extra herpes buffer to the inoculum. My fairly weak excuse was, well, but look at the color of the inoculum, it's still pink, so the pH didn't change much. But I mean, as as soon as you close the mouth, as soon as it mixes with the saliva. Yes, so probably, maybe that's a bit too early to say, but possibly a buffering capacity of your inoculum is a very big factor in how well it works with the oral application. Definitely something that we need to look into going forward. The second thing with the mucosal injury, I don't know. I mean, I agree, and Carolina can also comment on some of the feed studies that they did, where that may have an effect. We're also hoping to look into that with future experiments, for example, by mixing the virus with, I don't know, crushed walnut peels or something, where you have some rougher edges that would cause some lesions in feeding. But there's still a lot of controversy if you actually need these lesions or not.
So comment with the feed study, we did a totally different experiment where we contaminated feed to infect pigs, and what we found was that we could go down a log in dose if we separated it across 3 little portions separated by 10 minutes, rather than giving a higher dose in one portion. And I think that has to do with the contact duration, because we know that the virus needs to get in through these mucosal tissues in the oropharynx. And again, with this probability, if you can have less virus if you keep it there for longer. So I think that's some tweaks that we want to try with the IOP inoculation too, because we have the pigs sedated anyway. So instead of we do now 2 mils for 1 minute, we can do 3 separate bouts of 1 mil for 10 minutes and just keep the sedation for a little bit longer.
And another...
I can say it also did work last week when we did it.
And another addition to that is also feeding after the inoculation may have an influence, because that is something that Carolina never did. I mean, they feed them eventually, but not right after. Whereas in our case, because you don't want to feed them in the morning because you're going to sedate them, so you withhold feed until you're done with the inoculation, then you wait for them to wake up, and in our case, we then immediately fed them. And that may also not be optimal for the virus uptake.
Okay, sorry, thank you. We will take 2 last questions. Okay, 1 last question, but please be quick. Can you please raise your hand? Okay, thank you.
Thank you to all the presenters. 2 really quick questions. For Anne-Sophie, have you looked for virus in the pancreas of guinea pigs, because it has been described as a replication site in mice? And the other one for Michael, have you found any correlation between the strain of a virus and the success in the infection in pigs?
For the pancreas, no, we did not look into that. For the internal organs, we looked into the heart, the lungs, the liver, and the spleen. But that might be a good idea maybe for future experiments, that we also look into the pancreas to see whether there is some virus that can be observed there.
I think the easiest answer is yes. I mean, what we did last week was we used the very strains that Plum Island had used. That was an A24 and O1 Manissa, and really just other vials from the same prep. And we did get a lot higher incidence of successful infection after IOP inoculation with these viruses, but very small numbers. I mean, we're talking 2 pigs, 3 pigs, etc. The group in Japan that did use IOP a lot, they have a fairly long list of viruses that they tried, different O lineages, etc., always presumably with exactly the same method, because it was done by the same people over a fairly short span of time, and they also clearly have some viruses that worked and some viruses that don't. So there may be other factors at play as well, as we've discussed, but I do think that there certainly is some association with the viral lineage as well, yes.
Thank you very much. Thanks again for all your presentations. And we move on with the second round of presentations. Our next speaker is Lindsay Gabbert. Lindsay is a microbiologist with the US Department of Homeland Security Science and Technology Directorate at Plum Island Animal Disease Center. As a subject matter expert for the design of validation studies to assess virus inactivation, much of her interest has focused on filling critical scientific knowledge gaps related to depopulation, disposal, and decontamination. Lindsay will today present on inactivation of FMDV with hydrated lime solutions. The floor is yours.
All right, thank you for that introduction, and I appreciate the opportunity to speak with all of you today. So we've heard a couple presentations today from people at Plum Island. We have 3 federal agencies out there at Plum Island. I work for Department of Homeland Security. Part of our research focus is a little bit different than the other agencies. We have an applied agricultural biosecurity research program out there. Really the focus of that program is to provide actionable scientific data to support response and recovery phase of transboundary animal disease outbreaks. We look at virus elimination through cleaning and disinfection, survival in the environment, how do we best dispose of carcasses, do depop, that sort of a thing. These research projects are largely stakeholder driven. We work a lot with the industry partners such as National Pork Board, in addition to other federal agencies to support these projects. I'm going to give you the bottom line up front. I'm going to present the results first since I only have 8 minutes and I don't want to run out of time here. So basically the results of this project, we're looking at solutions of hydrated lime for inactivation of FMD at conditions found by pork producers in the field, including low temperatures and increased organic soil loads. And our results were that it actually works very well at ambient and under cool temperatures for inactivation of FMD, regardless of the soil load. It had a little bit of difficulty in activating FMD under freezing conditions, and that is what I will present to you next. Oh, and there's bonus results. Just keep in the back of your mind here, we did all of the tests that I'll present to you. Also with African swine fever, under all conditions, it worked just as effectively and sometimes better against ASF than FMD. All right, I'll tell you a little background of how this project came to be. Back in 2022, a group of US livestock producers reached out to us. There's various chemicals that are approved for cleaning and disinfection on farms in the US. However, most of these products are not tested against select agents such as FMD and ASF. So they came to us, they said, these are the products that we most commonly use on the farm. Can you please test them against these transboundary animal diseases and see if they're effective? So we went through this whole study. We produced these nice graphs for them, found new products that were not previously shown to be effective, presented this to them, and they said, great, but we don't care. If there's an outbreak, this is what our farmers are going to use. They're going to use lime. If chemicals aren't available, we want data to show that lime solutions will effectively work in case there's a shortage of resources. So we went back to the drawing board and trying to get to the next slide here. So in the US, hydrated lime solutions are used by many producers for standard routine control of endemic pathogens on the farm. However, there isn't a lot of data to demonstrate if they're effective against FMD. So our goal of this study was to determine the virucidal efficacy of hydrated lime solutions for inactivation of both pathogens under those conditions that they're going to find in the field. So what is hydrated lime? It took quite a bit of reading for me to really understand exactly what this test chemical was. It has a lot of different names in different countries. Basically, it's a solution, it's produced by mixture of quicklime, which is calcium oxide with water. It produces a calcium hydroxide powder. It's sold in the powder form. Once you mix it with water to create a solution. They call that slaked lime, whitewash, milk of lime. It's used in various sources, or has various uses, including wastewater treatment, neutralization of acids, water softening. Let's kind of be honest with ourselves. We know people have used lime solutions on farms for hundreds of years for control of pathogens. This project was basically to provide some quantitative data using more standardized, reproducible methods that other people can use to look at virus inactivation. So we do this through the use of a standardized quantitative carrier test method. The method we use is an OECD method. It's a standard method for evaluating virucidal activity of microbicides used on hard, non-porous surfaces. In these tests, we use FMD-A24. On LFBK cells, we had 3 different soil loads that we tested under 3 different temperatures: ambient room temperature, cool, and freezing. And then we're going to use a hydrated lime solution. It's about 40% powdered lime. This was a recipe specifically given to us from our swine producers that they use in the field in typical whitewashing applications. And we have a 30-minute contact time. You can see the picture there on the top right is the standardized stainless steel test coupons that we're going to dry virus on. So as I previously said, we're using about a 40% mixture of the lime powder with hard water that had a pH of about 12. That recipe, basically in the US, we can buy these bags of lime. They're about 50 pounds, 23 kilograms, sold in a lot of our agricultural or home improvement stores. So they just take those bags, cut open the top, and mix them in big tubs with water. We wanted to not only look at that concentration, but also to see if it's efficacious down to 1% as well. Our soil loads that we tested were a standardized OECD soil load that mimics mammalian secretions, excretions, so nasal discharge, that sort of a thing. We also looked at swine whole blood, and then heavy soil, which was a mixture of potting soil that was overlaid on top of the virus and the standardized soil load. We have 10 microliters of our virus plus the soil. On top of that, we're going to add 50 microliters of the hydrated lime solution. Allow it to sit for 30 minutes. The solutions are pre-chilled to each of the temperatures that we're testing, as well as the coupons, to simulate what we would find in an outdoor environment. Then we recover these samples in tissue culture media and assay them to determine CPE and TCID50 virus log reductions. So here's some of the results. You can see from the first set of untreated control columns there that regardless of the assay temperature, we get a good recovery of FMD, about 6 logs from all of our control untreated carriers. When you look at the virus dried in the standard soil load or in blood, it doesn't really, When you look at it dried in the standard soil load, we saw complete inactivation regardless of the temperature that we're conducting the test at. However, if you look in the heavier soil load, such as blood or the heavy potting soil, we see complete inactivation at ambient and cool temperatures, but under freezing conditions, we do see detectable live virus left over. This table pretty much demonstrates the same results as the graph there, but I thought it was interesting to look at the number of replicates that are actually coming up positive in those 2 treatment groups. So each time we conduct the test, we use 7 replicate or treated coupons. We conduct each test on 2 separate test days for repeatability. So you can see that most of the replicates there out of 14 are negative, except for the 2 bottom red ones, which are the tests under freezing temperatures in both the blood and the heavy soil. So even though we're seeing for blood a 99% reduction in overall virus, we're still getting infectivity in 11 out of 14 replicates, and then for the heavy soil, it's 9 out of 21 since we conducted that test a 3rd time as well. This graph is one of the more interesting findings, I think, from this research. So as I said earlier, we tested solutions from 40% down to 1%. When you're making a solution of hydrated lime in the field, especially for whitewash, you're using a higher concentration of that powder because you want to get a certain consistency that will actually stick to surfaces that you're applying it to. It's almost like a paint. However, if you create a solution down to 1%, it's kind of more runny and can be used almost as more of a disinfectant. That's not, the purpose of it is not to stick to surfaces. So regardless of the concentration, the pH of the solutions were consistent of over pH 12. And after 10-minute exposure at room temperature, we found that they all equally were effective for inactivation of FMD. We found no live virus, regardless of the concentration after 10 minutes at room temperature. We did not test each of those at the lower temperatures. However, I would expect at 4 degrees you'd see something similar, and again, at minus 20, probably less effective at the reduced concentrations. So the findings here kind of follow very common sense rules for disinfection. There's a sweet spot that chemicals are most effective. Generally, room temperature. If you get too cold, things freeze. If the environment's too hot, things start to evaporate. Obviously, clean surfaces are more easy to disinfect than those contaminated with high levels of soil. So this is no different. Follows all those traditional rules. We believe that the findings here demonstrate that if there's not commercial chemicals that are, available, that these hydrated lime solutions, even at lower concentrations, will be very effective for inactivation of both FMD and ASF. And I basically already said the conclusions. I'm not going to reiterate that. And that is the end of my talk. Thank you.
Thank you, Lindsay. Thank you very much. We now change our topic to lumpy skin disease, and I invite Nina Kresic, who will present on direct and indirect contact transmission of a clade 2.5 recombinant lumpy skin disease virus strain between cattle. Nina Kresic is a scientist at the Unit for Exotic and Vector-Borne Diseases at Cienzano. Her work is focused on the immune responses to experimental infections with capripox viruses and the modes of their transmission. Nina is currently engaged in developing and employing tools aimed at assessing both cell-mediated and humoral immune responses related to experimental infections and vaccinations. Nina, the floor is yours.
Thank you, Kirill. I'm very happy to be here to present today our work. I will briefly bring key aspects of lumpy skin disease and discuss modes of transmission, and then I will present the results of 2 independent animal trials that we did to investigate the transmission. So, lumpy skin disease is nodular dermatitis. It affects primarily dairy cattle and water buffalo. It can be presented as clinical disease in severe form, where you have multiple nodules affecting skin of the animal, or as a mild disease with only few nodules present on the skin of the affected animal. Very important for lumpy skin disease are subclinical infections when animals appear healthy, but they can be viremic or shed the virus via nasal secretion. The cause of this disease is lumpy skin disease virus. This is double-stranded DNA virus. Previously, LSDV strains, they were classified in 2 major clades, clade 1.1 that comprised mainly vaccine strains, and clade 1.2 with wild-type classical strains, which were causing infections in Africa, and from there they spread to Israel, to Turkey, and then in 2015 they even reached Balkan, Russia, and Kazakhstan. in 2017, for the first time, new recombinant LSDV strains were reported in the field. Today, they are clustering in clades 2.1 to 2.5. Since 2019, these strains are circulating also in Asia, together with clade 1.2 strains. They are important as for these strains, it was demonstrated something new, that they can be transmitted without vectors. So what are the routes of transmission for lumpy skin disease? The most known and the most investigated mode of transmission is vector transmission. LSDV was considered primarily as vector-borne disease, but today we know that this is typical for clade 1 strains, so classical wild-type strains. Vector transmission is achieved via blood-sucking vectors, such as mosquitoes or ticks, and they are primarily responsible for close-range spreading. If you transport the animals in incubation period or subclinical animals via vehicles to distant areas, then LSDV can be spread to distant areas, of course. Non-vector transmission, as I said, is something that is attributed to clade 2 strains. It can be direct or indirect. Direct transmission is achieved upon contact with secretions from infected animal. Today, we also know that intrauterine transmission is possible for lumpy skin disease, and nevertheless, indirect transmission, which is primarily achieved via contaminated fomites and iatrogenically during the veterinary procedures. For clade 2 strains, it was shown that non-vector transmission is something that is possible, and this raised concern as these strains are now circulating in previously unaffected areas. This is why we aimed to investigate clade 2.5 recombinant lumpy skin disease virus transmission to direct and indirect contacts in the absence of vectors. To do that, we did 2 animal experiments. We did them in our BSL-3 facility, vector-proof facility. So in the first trial, there were 8 inoculated animals. They were housed together with 6 naive animals. They served as direct contacts. And then we had additional group, which was in the same stable, but in the box, which was a bit distant from the other animals. And these animals served as indirect contact animals. It's not working. Animals were followed for 41 days in total. They were challenged with 2.5 clade recombinant LSDV. We used combined intravenous and intradermal inoculation. In the second trial, similar setup, a little bit less animals than in first trial, 5 inoculated animals, and then 5 naive animals housed together with them. They were followed for 30 days. They were challenged with the same virus. And during this time, we collected clinical scores, nasal shedding, viremia, seroconversion, and CMI response for these animals. So what are the results of our first trial for our inoculated group? Our infection model proved to be very successful. We got 6 animals with clinical lumpy skin disease. They were generalized lumpy skin disease cases, and 2 animals developed subclinical lumpy skin disease. First time we detected LSDV was around 3 days post-infection in blood, and by the 6th day post-infection, there was an animal with first nodules developing. Over the time, all animals demonstrated LSDV shedding in the nasal secretion. So the incubation period is around 6 days for the inoculated animals. The results for our direct contacts. All 6 of them acquired generalized lumpy skin disease. First time we detected LSDV in this group was 16 days post-infection. It was in nasal secretion, and by the 23 days post-infection, there were already nodules present. Animals became viremic, and by the end of the trial, all of them displayed clinical lumpy skin disease. So the time until the first detection of the lumpy skin disease in this group is 10 days after the clinical symptoms in our inoculated group. For the indirect contact group, here we again observed direct transmission of lumpy skin disease. One animal became clinically affected, developed generalized lumpy skin disease. The first detection was around 23 dpi, again in nasal secretion, and the first nodules became apparent at 30 days post-infection together with viremia. So the time until the first detection in this group is 17 after the first clinical symptoms in our inoculated animals. In the second experiment, similar findings were observed. We got less clinical cases, 2 animals became clinically affected, and 3 animals were subclinical. The incubation period was again similar. At 7 dpi, we observed clinically affected animals. For our direct contact animals, 2 of them, they again became clinically affected. 1 animal was subclinical. So LSDV was again transmitted to direct contact group. 10 days after the first symptoms in our inoculated group, we observed first clinical symptoms. For the seroconversion and CMI response in both trials, for the inoculated animals, they all seroconverted. In first experiment, direct contact animals also seroconverted, including indirect contact animal that was affected. For the CMI response, all animals in both trials developed measurable interferon gamma levels, regardless of the clinical form of the disease that they were having. In conclusion, clinical outcome after the challenge of cattle with a clade 2.5 was highly similar what was observed for classical strains from the clade 1.2. Non-vector transmission of clade 2.5 LSDV was shown to our direct and indirect contact animals. That was shown in 2 independent animal experiments. And this is important because these findings can impact LSDV epidemiology, mostly because these new modes of transmission, they are vector independent, so they can be the disease can be transmitted outside the vector season. And now it remains to examine the efficiency of different transmission routes. I would like to thank all of my colleagues for this important and exciting work, and thank you all for your attention.
Thank you very much, Nina, for this presentation and very indeed relevant findings for the prevention and control of lumpy skin disease. Our last speaker for the session today is Andy Haegeman. Andy has been working with Cienzano since 2003, with the last 13 years in the Department of Exotic and Vector-Borne Diseases. He is involved as a molecular biologist in various research projects and routine diagnostics on blue tongue, capripox viruses, Rift Valley fever, parapox viruses, and FMD. He focuses on the development and validation of diagnostic assays, vaccine evaluation, and transmission of capripox viruses. He also provides support and supervision for international capripox training courses and various activities of the EU RL capripox viruses. Today, Andy will present on lumpy skin disease vector transmission. Are the numbers important? Andy, please.
Thank you, Kirill, for this nice introduction. It left me almost speechless, but I will still try to fill my 8 minutes trying to explain my presentation. Lumpy skin disease, vector transmission, are the numbers important? Now I was lucky that my colleague Nina went before me, so I don't have to explain lumpy skin disease anymore and the transmission pathway. She did it very nicely. To summarize, you have non-vector-borne transmission, which has been demonstrated by her, that this mainly is associated with the recombinant 2.5 strains and less for the classical wild-type 1.2 strains. For both of them, of course, vector-borne transmission remains important, and it has been shown in the past to be a really important factor in virus epidemiology. So what did we know about vector transmission from lumpy skin disease? Well, for example, it can be transmitted by ticks, by our South African colleagues. Goatpox, which is a family member of lumpy skin disease, has been shown to be transmitted by Stamoxis, the stable fly. And for us, this was very interesting because the stable fly is very common in many countries, widespread. So we were very interested if it was possible. And indeed, we did, with a few trials, demonstrate that LSDV can be transmitted by the stable fly in vivo and in vitro feeding. But what all these trials had in common in the past was that we use a lot of vectors to achieve this transmission. For example, I will come back to that in a few slides. When we do the transmission studies, we use several batches of flies. We feed them multiple days and transfer them to the acceptors. We use like 350 flies per cow, multiplied per 3 days, and keep the math simple. Let's say 1,000 biting events that you have. This is a lot. But of course, this is relevant because if you look at the field, and this is a picture from a cow, you always have multiple flies around the cow. There's not one stalker fly who follows one specific cow everywhere. No, you have a lot of flies. But is this important? Is it important that you have a lot of flies there that can bite and transfer to another animal? That's why we did a new trial where we took 10 donor animals. We challenged them, like we challenge them always, by a combination of intradermal and intravenous inoculation. We followed different parameters, but in this case here, viremia was very important. And 5 from the animals in this trial became viremic. And that was a time point that we go to the field and we capture some Stamoxis from the field and bring them back to the stables. From this point, we separated the trial in 2 different components. We had, again, a mass exposure group for 5 animals, and that's what I mean by giving them a lot of flies. So 2 batches were caught and placed and transferred to the acceptors. Each batch was fed 2 to 3 days. The 300 flies, 1,000 biting events. So this was our control group because we knew in the past this always worked. So this we used as a control for the cows, for the flies, for the virus, for everything. But we now included a second group of 10 animals, where we call the low exposure group, where we give them only 1 cage with 20 flies 1 time. So with the simple math, let's say this is 20 biting events. So this is lots less that we have here. Now what are the main results? I will keep it short for the 8 minutes. This is a small overview of the clinical signs of these 10 animals with a low exposure. Here the flies were transferred to the naive acceptor animals, and you see here 4 animals developed clinical signs, so the nodules, the fever, and all that. There was a 5th animal which became clinically much way longer. Is this a delayed incubation period, or is this maybe the first evidence of non-vector transmission? That I don't know for the moment. But these 4 were from the limited exposure to the flies. When we now look to the viremia, we see the same picture. You have 5 animals becoming viremic, and one of the 5 is a late onset of the clinical signs. Same picture again for virology, where we see on the immunoperoxidase monolayer assay, 5 animals, you had the 5 animals which were clinical and viremic seroconverted, but interestingly, we had a 6th animal which seroconverted without showing any clinical signs, without any viremia, but it did seroconvert, and it also did on the ELISA. So what can you conclude from this experiment?
that when there are low exposure, so minimal number of flies, 5 out of 10 became infected. Became infected because they became clinically ill with the nodules, the fever. They were viremic in the blood, seroconverted for the antibodies, and these 5 animals is 50%. And this was almost the same percentage after mass exposure, and even the same percentages as we inoculated the animals. So that was a little bit surprising for me. I didn't think that would happen. One additional animal did seroconvert without showing clinical viremia or clinical signs. So we can say transmission confirmed by a few flies. So now we know that transmission can happen with a lot of flies and with few flies. And for us this is important because it has several important impact factors. You have conditions where you have less vectors, like for example, if you have transport, you're transporting your cows, a few flies can come in or come out the trucks, so these are not without any danger, certainly if you pass through a free area. Vector control, you have to be really efficient to be able to protect your stable, because even if you have a few flies, it can cause an infection. And we have to think about also the vector seas that we're always talking about. Even outside the ideal temperatures and humidities, and you have less vectors there, it still can be an issue. So I did not do all that work myself, of course. I have a fantastic team from me with Wannes and Nina, they are here, and David. Of course, our technicians who are working in the lab, and of course our team in animal facilities, which do the experiments and the sampling and the clinical scoring. And I thank you for your attention. And I hope I was in my time, Kirill.
Thank you very much. Andy was perfect, and thank you for the concise presentation. Now we still have some minutes for a set of questions. I would also like to invite Lindsay to join us. So please, we are open and waiting the questions from the auditorium. While we wait for the first one, maybe Nina, I have one question. It was very interesting to hear that you already proven the non-vector transmission of clade 2.5, but how much we know about the non-vector transmission of the clade 1, which was, as you mentioned, present in the Balkans and spread there. Do we have some data to support similar conclusion on this, or any comment on this?
Well, I'm sure Andy can add a lot of to this question. I will try. Direct non-vector transmission for classical clades, for classical strains from clade 1.2 is considered highly inefficient. So, but I know that there was a study in 1995 that investigated whether direct transmission was possible, and their findings supported the inefficient transmission route. That's pretty much it from me, but maybe Andy from his experience with classical strains can add more.
Well, I can confirm what Nina said. We haven't been talking about our colleagues in South Africa where the disease is more endemic, and there they don't see much direct contact transmission. That's one thing. I think there was also a study from Russia where they've used infected feed, bread, and where they tested the 1.2 and the 2.5, and they saw transmission with the 2.5, but they did not see it with the 1.2.
Yeah, that's very interesting. Thank you. Do we have a question from the audience?
Thank you very much. Very interesting studies. Andy, just a question. You mentioned the viremia in the donor animals for the then fly transmission. We did some studies a few years back where we found out the uptake from stomoxys of lumpy skin disease is much better when they feed on the nodules than when they just feed on skin, even if they also have a viremia. So did your donors also have nodules, and have you seen the same, that that the uptake of the flies is much better when they directly feed on lumpy skin nodules?
In this trial, they were not specifically fed on the nodules. They were fed on the shoulder area. There were that time no nodules because the viremia in this trial was a little bit earlier. Nodules can appear a little bit later. And we saw a viremia, and we said, okay, we'll not take the chance to lose that viremia. We'll directly place a nodule on that. If the uptake is better on nodules, that we did not test or use. But from my experience, and also because we did a lot of feeding of Stomoxys in the lab, Stomoxys will feed on anything. So it will take up virus very, very, very easily.
Thank you. We have one question here and the last one there. If we have time, Michael, for sure.
Hi, Andy. So my question is, when you show that doses don't really matter with the amount of insects, what do you think about doses of actual virus when we inoculate so much virus intravenously? Would we see the same thing if we used, 1,000 less virus intravenously? Would we see the same thing? Or what are your thoughts?
You do see a difference in speed. We use now this amount because it's standardly now done in our model. We have an idea when the viremia more or less will occur because you have to coordinate with the capture of the flies in the field. That makes it tricky that your viremia is at the same time that we have good weather in Belgium, which is not always easy. But of course, if you change the doses, you will change the course of the disease. And that way, it influences certainly the viremia and the onset of the symptoms.
What about the amount of animals that get infected? So again, it's always, you only get half the animals, or it's variable. It's the same thing that use a lower dose.
Well, we did a few trials with lesser dose, but very limited trials, maybe one trial per dose. And from one experiment, it's difficult to say, okay, this percentages we always have. So I did not do enough repetitions to be sure, okay, the average amount of viremic animals will decrease. That kind of data I don't have.
Thank you. And one question from a colleague there.
Thank you. The team from that worked on LSD, just to ask the last presenter, 20 flies, 350 flies, did you confirm the presence of the virus in each of the Stomoxys that you put in the population for infection?
We did test a few Stomoxys per case. In this case, we had only 20 Stomoxys. I think we tested a few, but not all.
Okay, thank you.
No, yeah, because my concern is if you do not test or confirm the presence of the virus in each of these fly, the chances are that the virus load that might come into the host might not be what you're expecting ab initio. Just put that into cognizance.
No. For that I agree. But the fact what we did is, on the moment that we place the cages on the animals, we monitor the animal's response because they become agitated the moment that the flies bite. And then we transfer that cage to the acceptor animals and let them bite a second time. Of course, how much each fly takes up and how much is transferred, that we do not know. But we did know that the animals, 50 of the animals at least became positive. So we know that it is possible to get transmission from a few flies. Maybe why some of the other flies, other animals did not become clinical, it's possible that's due to a difference in transference of the virus. But okay, we just wanted to see if it was possible, if you have only 20 flies in your cage, that you get transmission.
All right, thank you. So to the person from DHS, yeah, hydrated line. I want us to look at it from a very realistic perspective here. In resource-poor countries of the world, LMICs. Most of the cattle you have are always owned by nomadic people, herders, and they transit from one point to the other. So even when you have outbreaks of FMD, reportage becomes a problem. Even identifying the herds affected, it's always a problem. Now, how applicable is the use of hydrated lime in such scenarios?
So if I heard your question correctly, you're asking in scenarios where cattle are grazing outdoors and often moved around quite a bit, how that solution could be useful in that situation, correct? Well, so the hydrated lime as a whitewash, I would have to personally have a better understanding of exactly how that process works. Are the cattle always in the field? Do they come back to eventually to a certain location where they're held, or they're just grazing animals? So, yeah.
It's a comment.
What we have in realistic context, especially in resource-poor countries, most of these animals transit in search of pasture and water from different regions, and in the process, they might pick up this virus and cause outbreaks. So even if they get established here today, the next week you won't find them there. They migrate.
Yeah.
Then where do you apply this hydrated lime? Is it at the points of the outbreak where you don't find these animals and where they might not come back to until the following season of raining season for greenhouse pasture, or is this lime also applicable on the animals themselves as they migrate?
Yeah, I would say that it's not specifically recommended for use on the animals themselves. It's more on the fomites, on permanent structures that you may see in the field where it can be applied as a whitewash. Also, historically, as a whitewash solution is applied to surfaces allowed to sit there, and it continues to allow for pathogen reduction over time. So once it's on there, the pH remains fairly stable. So if new pathogens are introduced, it continues working. So it's not just a one-time 30-minute application.
Thank you very much. I think we are running out of time, and I would once again like to thank our speakers for today, and thank you for joining us for this session. Before we break for lunch, few practical informations. I'm sure that you were able to see the list of posters relevant for this session, and you can see the posters in the reading coffee room. As for the lunch will be served at Refectorio, Galleria Refectorio, and St. Thomas. But an important point for all people with any dietary requirements, they need to go to the restaurant St. Thomas. For the others, we have the other two restaurants. Thank you once again, and we will reconvene with session 3 in this same room.